So we're going to have 3 classes on the mechanisms of different parts of the virus replication cycle. So today we're just going to be looking at the attachment and entry. So every virus, yes. Oh, okay, I'll try and correct afterwards. Sorry, my mistake. I uploaded the wrong PDF. So yeah, so in order to replicate, every virus has got to go through the replication cycle, and it's really this kind of framework is true for every single virus. Okay, so first they've got to bind to the host cell somehow, then they've got to get into the host cell. Just staying attached to the surface is not enough. They have to enter the host cell. And then at some point, the virus genome has got to be released from the particle. So this is called decapsidation. Then after decapsidation, 2 things have got to happen. The genome has got to be replicated, and viral genes have got to be expressed to produce all the viral proteins. And then once the infected cell has produced a lot of copies of the virus genome and a lot of the structural proteins, then new particles can be assembled and they've got to be released. So all of these steps have to work in order to go from, 1 virus to 5 viruses released from this infected cell here. And if you can somehow block one of these steps, then you can stop virus replication. So this is the basic idea behind any antiviral drug therapy. If you can get a molecule that can block one of these steps, you can inhibit virus replication. Yes?
How the virus go outside of the cell with this cover?
Okay, how does the virus escape from the cell? For non-enveloped viruses, 2 mechanisms, lysis, and also they can be released in a kind of pseudo like membrane, but that's for like so this is a question for in 2 lectures. So today we're going to be concentrating on the first step, which is like attachment and entry. Next class will be expression replication. The final one will be assembly and release. So attachment and entry, the first step is always mediated by a specific interaction between a virus attachment protein and a host cell receptor. And these interactions are often low affinity, but you have a lot of copies of the viral glycoproteins or the attachment proteins on the capsid on the surface of a virus. So you have multiple interactions on the same particle, and that ends up giving you something which is high avidity. So each individual interaction is low affinity. Okay, so the nature of the host receptor is something that plays an important role in virus tropism. That means which cells can be infected and which species can be infected by this particular virus. So if we just look at a few examples for enveloped viruses up here and non-enveloped naked viruses down here. Then, for example, HIV, the attachment protein is GP120, binds to the CD4 receptor, which is expressed on CD4 positive T cells, dendritic cells, monocytes, macrophages. So that explains why this virus infects T cells and why it causes the disease that it does. Because once you've destroyed all your CD4 T cells, you don't have an effective adaptive immune system anymore. Similar thing with Epstein-Barr virus, which, as we all know, is now officially called lymphocrypto human gamma 4 virus. So, okay, So its main receptor is CD21 on B lymphocytes. Okay, so this cell is going to infect B cells. And it's why it's related to B cell leukemias. Okay. SARS-CoV-2, spike protein binds on a protein receptor on epithelial cells. For non-envelope viruses, okay, there are no glycoproteins, so it's the capsid proteins that, yeah. Okay. Well, I was going to come back to this one, but okay, yeah, probably I should have said this in order. So still staying with envelope viruses, okay, we've like most of these receptors, or the ones that I've listed anyway, they're membrane proteins, but viruses often use glycan receptors for attachment. So that's the case for influenza viruses. The attachment receptor are surface glycans with sialic acid. So these are pretty much available on all types of cells, okay, all types of eukaryotic cells, or human cells anyway, vertebrates, shall we say. So for influenza virus, the attachment receptor has got a wide distribution, but mostly its replication is going to be restricted to the respiratory tract. And for this virus, it's not the distribution of the receptor that is responsible for its tropism, okay? Often it's the receptor expression which determines which organ, which tissue, which cell type can be infected, but not always, okay? And we'll see why later on. So some receptors are glycans. That's the main thing here. Capsid proteins on non-envelope viruses, either it's a single capsid protein, which can form the binding site, or it's a kind of association between different capsid proteins. So for example, if you imagine poliovirus, you've got like an association between EP1, 2, and 3. The receptor binding site is kind of here, where they all meet together. Okay, it's not on one individual protein. And once again, non-envelope viruses, a lot of them can use glycans as binding receptors. Once again, also here, this is another example where the binding receptor is not the only thing involved in tropism for adenoviruses. Yeah. Okay, good question. Okay, so I've got sialic acid here twice on a slide, and here I've got a specific tropism, and here I have no tropism. And the question is, why is that? They're over there. So the answer to that is sialic acid, it's an oversimplification. Okay, so when I was learning biochemistry, I do remember I had a lot of trouble with glycan structures. Proteins is kind of easy, it's linear, you've got 20 different amino acids, one after the other, that's okay. But the glycans are horrible because, you've got a bunch of different sugars and they can go in different links from one to the other. So if you remember N-links glycoproteins, for example, you'll have some kind of base structure here, which I don't quite remember, and then you'll have mannose, and then maybe with some branches with mannose, and then these get decorated with often with sialic acid, okay? Maybe something else. And so the binding receptor for influenza viruses is going to be sialic acid, mostly N-linked or O-linked, I'm not really sure about this, but on surface glycoproteins. And the thing that it's going to like is, yeah, an alpha 2 6 on alpha 2 3. And there's a bit of a difference between avian and human influenza viruses here. So for seasonal influenza viruses, they prefer to bind to sialic acid with an alpha 2 6 linkage, and avian influenza viruses alpha 2 3. This is going beyond a bit what I wanted to talk about, but since we're on the subject, okay. So in the human respiratory tract, up in the upper here, you know, in your nose, upper respiratory tract, we've mostly got alpha 2 6 sialic acid on glycoproteins. If you go down in the lungs, okay, the lower respiratory tract, you also have alpha 2 3. So this means you can get infected by an avian influenza virus, but the virus has got to arrive here, okay? But when you get infected, most of the time, all the aerosols or particles get deposited in the upper respiratory epithelium, okay? Just because of the way the airflow goes when you breathe. So infections are rare with avian influenza viruses. Okay, so that's influenza virus. And what about BK polyomavirus, which I know everything about, fortunately. So for BK polyomavirus, it's sialic acid, but on another type of receptor, which are gangliosides. So gangliosides, they are kind of glycolipids. So you have ceramide here, and then some kind of sugar down there, maybe galactose. And then, of course, you have different branch structures again, which are different from one ganglioside to another. So what the BK virus likes is sialic acid bound to another sialic acid in an alpha 2 8 linkage on a ganglioside. Okay? And you only really have this in the kidney and also a little bit in the brain. Okay, so there's a bit of a long answer, but the answer is sialic acid, okay, is just the terminal sugar, but of course it can be involved in different glycan structures, and these glycan structures are different for the two viruses. Here, like widely distributed, here with a specific, distribution only on certain cell types. Okay, so one of the things is how do we know all of this stuff? And no, for a long time, the first thing to do was the first question you might have, okay, is my receptor a protein or some kind of glycan? So what you can do is get the cells that are susceptible. Viruses can bind and infect the cells, and you can treat them with proteases or glycosidases. If the cells can't bind anymore after protease treatment, then your receptor is a protein. Okay, so it gives you some kind of information, and then look at whether the virus can bind and infect cells. So more and more, this is being complemented or replaced by a molecular biological approach, and there's two ways to do this. Okay, so the first way is you get a cell or cell line that cannot be infected. And it's got to be permissive, but non-susceptible. So these are technical terms that have a very specific meaning. So a susceptible cell means the virus can bind and can enter. Infectious entry is possible. A permissive cell, the virus can replicate if it gets inside the cell and release new virus. So in order for a full replication cycle, the cell's got to be susceptible and permissive. If it's only permissive, the virus can't get inside. If it's only susceptible, the virus can attach and enter, but it can't release new virus. Replication cycle doesn't work. So what you're looking for this type of experiment is a cell line that's non-susceptible, but still permissive. So it's lacking something for the attachment and the infectious entry. And then you kind of transfect this cell line with a library of different cDNAs coding for different proteins. And your idea is one of these proteins is going to allow the virus to infect my cell. So I have to find the clone where I've got the cDNA that allows the infection to happen. And then you pick up that one and you find out what gene was coded on your cDNA library. So that's one way. You start out with a cell that can't be infected and you look for the gene that allows it to be infected. And the opposite approach is this. Now we can use CRISPR-Cas9 to knock out every gene in a cell population. And this time you want to look for the knockout that prevents cells from being infected. And this works especially well if your virus is cytopathic, if it's lytic. Every cell that gets infected is going to kill it. So if you knock out the gene that allows the infection to occur, then those cells will survive. So you just have to collect up the surviving cells and find out which gene got knocked out. Okay? So this is kind of and once you do this, you can say, okay, I've got, this molecule is my receptor or co-receptor or something. Okay, so we do a bit of this in the lab with the biochemical approach. So as I said, I know everything and probably more than is healthy about BK polyomavirus. So like I said, it depends on sialic acid for infectious entry. And the enzyme that removes sialic acid is called neuraminidase, because sialic acid, another name is neuraminic acid. So we've got cells infected with the wild type pseudovirus. You can see the infected cells here. If I treat them with the neuraminidase, I don't have so many infected cells. And if I look at the binding, this is the non-treated cells. I've got nice binding with this wild type capsid. If I treat them, I don't have any binding. So I've got sialic acid dependent attachment and entry. So in the lab, we got this mutant that was growing in a patient called the NQ variant, just had 2 amino acids change. And this one, you treated the cells by neuraminidase, doesn't really change. In fact, you get more infection. And if you look at the binding, it's just the same with or without neuraminidase. So we're kind of quite intrigued by this. So there's another receptor for this variant. And in fact, we found out what it is. So we're kind of writing this paper up to explain what the new receptor is. Okay, so those are receptors. Now, for the entry process, I'm going to talk about, for example, one big classical example for an envelope virus, and then later on, and then try and generalize what we saw to other envelope viruses. And then I'll go on and give one example of a non-enveloped virus. So the big classical example for envelope viruses is influenza, which is part of the Orthomyxoviridae family. Now, the Orthomyxoviridae can include A, B, and C influenza viruses. So influenza A and B can infect mammals and birds. Influenza C infects only mammals, affects humans, but it's mostly mild or asymptomatic. So there's not that much research on influenza C. Influenza A and B viruses cause more pathology. They make you sicker. So for example, the vaccine against seasonal influenza, it's a mix of influenza A and B serotypes. There's no C. Virus, these viruses are quite different. They are transmitted by ticks. Okay, so they're not respiratory viruses, they're tick-borne viruses. So looking at the structure, the Orthomyxoviridae, they are enveloped viruses with a helical nucleocapsid. And the organization and the genome is a negative strand RNA genome. So it's a Baltimore class 5 virus. And what is quite specific about the Orthomyxoviridae is the genome is segmented. Okay, so each virus particle will contain 8 different RNA molecules associated with the nucleoprotein. And basically, each one of these segments codes for one virus gene. So for attachment and entry, it's going to depend on the virus glycoproteins. And there are 2 viral glycoproteins, well, 2 major ones, the hemagglutinin and the neuraminidase. And the attachment and entry protein is the hemagglutinin. So the overview of influenza virus attachment and entry is first they'll be binding to sialic acid on glycoproteins. These particles will be internalized by endocytosis. Then the maturation of the endosome causes the pH to go down. So you get acidification of the endosome, and it's this which is going to cause membrane fusion. So membrane fusion is where you go from this situation to this one. So here you've got the virus and the viral envelope attached to the endosomal membrane. And here you you're supposed to be able to see that they become continuous. So this forms a pore, and all these ribonucleoprotein segments, they can get out. So this is decapsidation, which is occurring here. So, okay, so the virus genome has been released from the particle. That's decapsidation. So that's the general process. How does it work? So we have to look a bit closer at the structure of hemagglutinin. So the active form is a trimer, and this is a look at one monomer of hemagglutinin. And there are 2 domains, HA1 domain and a HA2 domain. And it's a type 1 membrane protein. So the N terminus is outside. Is that type 1? Oh, you know this membrane protein, C terminus inside, outside or something. Okay, so the N terminus is up here and the C terminus is down there. And there's a transmembrane region in HA2 which anchors the whole thing in the viral envelope. So HA1 is a globular domain. That's the receptor binding domain, and HA2 is these alpha helices, which is like forms a stem. So HA1 has got the binding site for sialic acid here. So what happens during attachment, well, during membrane fusion? So this structure you see here is called the pre-fusion structure. So you've got one alpha helix comes up here, and then as you follow the strand round here, it goes back down. And then at some point, you've got at the end this fusion peptide, which is over here, which is kind of buried down at the base of the stem. So you have these 2 alpha helices stacked up kind of like this, one next to the other. Now, when the pH is reduced in the endosome, what's going to happen is you've got the hemagglutinin trimer like this. Up here, you just have a representation of the HA2 domain. One alpha helix going up, one coming down, and the fusion peptide kind of in yellow down here. So the first thing that's going to happen is that the HA1 domain is kind of getting detached here. It's pushed to one side. And then the next thing is that these alpha helices are going to suddenly expand. So instead of being one up, one down, it goes like this. So it's kind of like you've got a spring that's compacted, and then all of a sudden you release the tension. So the spring gets much longer. And this will project the fusion peptides into the endosomal membrane. Okay, so you've got this structure which is suddenly going to get longer. It's going to go and insert these fusion peptides into the membrane. So this is the elongated or the extended structure form of the structure. This is unstable. So the next thing is going to happen is going to collapse or fall back. And as it falls back, then you'll have the endosomal membrane being brought really close together to the viral envelope. And then because you have all these hydrophobic peptide domains here in close proximity, this will lead to membrane fusion. And at the end, your fusion domain is completely folded back on itself with the fusion peptides also inserted into the membrane here, along with right next to the transmembrane domains. And this is where you finally get the pore. So this kind of organization with the N-terminal domain being involved, being responsible for the attachment, and the C-terminal domain with an alpha helical type 1 fusion domain structure is something you find in a lot of different envelope viruses. So for influenza, it's the hemagglutinin, HA1 is the attachment domain, HA2 is the fusion domain. For HIV, GP120 is the head domain, GP41 is the fusion domain. For SARS-CoV, well, all coronaviruses, it's a spike glycoprotein with the S1 domain responsible for receptor binding, so often called also the receptor binding domain, and S2, the alpha helix. And in terms of structure, you can kind of see it. Okay, so each time there's a trimer here. So you can see, you know, the receptor binding domain up at the top, receptor binding domain here, receptor binding domain up at the top, and then the alpha helices, they're down at the base. So similar kind of structure, and they're all going to work in the same way. So one of the things not to forget, yeah.
Does the alpha helix recognize the receptor?
No, the alpha helix does not recognize the receptor.
How do they fix to the cell membrane if they don't recognize?
It's just because it's a physical chemical thing. The peptide is hydrophobic. So when it's like projected up like this towards the lipid membrane of the endosome or the cell plasma membrane, the hydrophobic peptides are looking for a low energy situation, right? And so they can like insert into the lipid bilayer. It's not a specific receptor, it's a physical chemical interaction. Yeah. The process is starting with interaction between the attachment domain and the receptor. And that's going to start a chain of events which will lead to the membrane fusion domain being activated or being triggered. Yeah, so if you don't have attachment first, none of this can happen. Okay. So what I was going to say, so these are examples where the attachment and the fusion functions are on the same protein. But in some viruses, they have two different proteins. Okay, so for measles virus or respiratory syncytial virus, you have a hemagglutinin protein, which is H. This is the attachment glycoprotein. And the fusion protein, F, is in a different glycoprotein. Okay, so you have a separation between the two functions. Okay, so HIV, coronaviruses, influenza, all of their attachment and fusion glycoproteins have pretty much the same structure and they work the same way. And they are examples of type 1 fusion proteins. And they are not the only ones. So as I said briefly yesterday, there are 3 types. Type 2 are formed of beta sheets. The functional subunit is a dimer, and they're basically flat against the viral envelope in their pre-fusion form, whereas type 1 fusion proteins or fusion domains are perpendicular to the envelope. And there are also type 3, which you find in herpes viruses and viruses like rabies virus. Okay, so I'll just go through these videos because they're probably going to be a little bit better than like this kind of thing, a little bit more dynamic. So I'll kind of open these ones if I can, yeah. So just to see how this works. So here's your coronavirus particle with spike trimers on the surface, and on the inside you have the heliconucleocapsid. So the first thing that's going to happen is going to be the binding to the receptor with the S1 domain. So these kind of like globular heads. Oh, what am I going to say here? Oh, okay. What are all these little gray things up here on this animation? No. Good idea. No. All of this whole thing is a spike trimer, okay? The S1 domain is this bit, okay? This kind of big club, like a big club. And the S2 domain is all of this long, thin part, the stalk. Okay, but what are all these little gray blobs there? What is that going to be? Yes, glycans, glycoprotein, right? So they've added the end-linked glycans on the glycoprotein. Okay, so that's what they are. So this is the interaction between S1 and the ACE2 receptor. This is the first step, receptor binding. And then in the case of SARS-CoV-2, what is required to activate or trigger the fusion domains is a proteolytic cleavage from this guy here, which is a protease called TMPRSS2, which will cleave at the what's called the S2 prime site. So S1 is removed. Look at that. It's physically detached. And that's what's going to allow these S2 domains to deploy. So they're going to go to the extended form, and the fusion peptides bury in the plasma membrane, and then the whole structure here is very, very unstable. So it folds back on itself, and then you get the envelope and the cell plasma membrane which fuse. And I like this part as well, because you can see that the virus envelope kind of dissolves into the plasma membrane. Okay, it's out here, and it kind of just goes like that. So everything that's in the particle has been released here. Okay. So that's the way this one works. So that's a good example of membrane fusion coordinated by a type 1 fusion domain. So for dengue fever virus, there's a pretty old animation now, but I still think it's really very good. So here we go. Here's the particle landing on a cell, and you can see that all the glycoproteins, they're lying flat on the surface. It's internalized into an endosome. Here, what's going to happen, the trigger is going to be reduced pH. So this is all the acidification, and you can see that all these envelope glycoproteins, they're kind of like this, flat on the surface of the envelope, and then they all stand up like that. And on the tips of my fingers, this is where the fusion peptides are. So the hydrophobic peptides in the pre-fusion form, they're all kind of interacting with each other like this, the hydrophobic peptides. But then when they stand up, they're available to insert into the endosomal membrane. So that's what they're going to do. They're going to insert into, so this is the virus envelope. This is the endosome membrane. Once again, the extended form is unstable, so it folds back, and this folding is what's going to bring the two membranes together and allow them to fuse. So this is a kind of imaginary vision of what is happening with these legs unfolding. But this is the idea, is you get the formation of a pore, and then once you've got the pore, all of this stuff, which is the virus genome, can get out. And it gets into the cell cytoplasm. Okay, so it helps you visualize what's going on here. Okay, so for type 1 and type 2 fusion domains, and also for type 3 fusion domains, the process is going to be quite similar. So all of them on the surface of the virus particle, they're in a kind of pre-fusion conformation where the whole fusion domain is compacted. Then there's going to be some kind of triggering event which causes it to extend, going to a kind of an extended conformation here. And this extended conformation is unstable, it folds back, and then the zippering is what's going to allow the pore to form. So all of these different fusion domains work in the same way. And there's a common framework for how they function. So the first thing is that there's going to have to be some kind of priming step. So when the fusion protein is synthesized on the ribosome and incorporated on the surface of the particle, it's not immediately active. That's important because you don't want the fusion domains to start working inside all the intracellular compartments of the infected cell. That would cause a huge problem. So for effective release, you don't want the fusion domains to be active straight away. They have to be primed first. So they're synthesized in an inactive form, and then they're primed. Then after the virus binds to its receptor, there's a second stimulus, which is required to trigger the membrane fusion. You get extension, then fold back, then zippering. So all of these fusion domains, they kind of work like this. So what is the priming step? So for a lot of viruses, I'm trying to think of an exception actually, but it's very often the case that the priming is a proteolytic cleavage. So for all of these type 1 fusion domains, and this one type 2 for dengue fever virus, the membrane and the envelope glycoprotein is synthesized as a kind of precursor. So influenza, it's HA hemagglutinin 0, it's got to be cleaved into HA1 and HA2. For HIV, GP160 is got to be cleaved into GP120 and GP41. SARS-CoV-2, all the coronaviruses, the spike glycoprotein is coded on one gene, it gives you one protein, but it's got to be cleaved into the S1 and the S2 domains. And the same thing for the dengue virus, the envelope glycoprotein is first synthesized as a polyprotein along with the capsid. I think this is precursor or something, and matrix and then envelope. And they've got to be cleaved to separate them out into the mature proteins. So sometimes these proteases are ubiquitous. So every infected cell will be able to produce a primed functional glycoprotein. But sometimes the proteases are restricted to certain tissues. So that's the case for influenza, okay? The receptors are widely distributed, but the protease required for priming of the hemagglutinin has got a restricted distribution. So that's why active virus replication is only going to be restricted to the respiratory tract. So it's not the receptor, it's the priming enzyme, which is the limiting factor, which is related to virus tropism. Okay, triggering, what are the, yeah. In a general case for virus particle maturation, can be a viral protease sometimes, yes. But for virus glycoproteins, I think it's always going to be a cellular protein. Why? Because this has got to occur in the, it could be viral, but I don't think there's a single exception, but you have to think about, where this is occurring. So here's our cell, here's the endoplasmic reticulum, and here's the Golgi. So the virus particles, they're either going to be formed, they're going to be released either from here, okay, or they're going to be released from here and mature somewhere here and be secreted, envelope viruses. So the enzymes that can do this, okay, they've either got to be, and in any case, the glycoproteins, they're going to be produced here anyway, so they're going to be like here. So forget about that. So the glycoproteins are going through this pathway. So all the enzymes that can act on them, they've got to be in the lumen of the endoplasmic reticulum or in the Golgi somewhere, or maybe outside the cell, but that's less likely, I guess. So there's nothing, I guess, to stop a virus from secreting a specific protease to do this, but I don't know of any examples. So it's cellular proteases because they're already there in this compartment. Okay, so that's the priming, triggering. Okay, we have to ask Vincent Macigno this one again, see if he answers. I'm sure he'll be very happy. So triggering, what is the event that really causes the fusion domain to explode or to fire off? So decrease in pH. Viruses that are first endocytosed, it's the low pH of the endosome, which is the trigger. Influenza, flaviviruses. What's another thing we saw for SARS-CoV-2? We saw it's the proteolytic cleavage. So as we saw on the video, there's the cleavage by this enzyme called TMPRSS2, which is the trigger. So that's true for SARS-CoV-2, but for other human coronaviruses, respiratory coronaviruses, often the proteolytic cleavage is caused by cathepsins, which are also in the endosome. So there's kind of two entry pathways for coronaviruses. TMPRSS2 is going to work at the plasma membrane, and the cathepsins are working at the endosome. And depending on the strain or the type of coronavirus, you'll have either a mix of both or one or the other, which is a majority. So there was something a little bit different about SARS-CoV-2, which was it was mostly entering at the plasma membrane by this TMPRSS2 cleavage. The other thing that can cause triggering is interaction with a co-receptor, and that's true for retroviruses. HIV-1 binding is to CD4, and triggering is by the interaction of GP120 with a co-receptor, the chemokine receptor CCR5 or CXCR4. So those are the three kind of triggers we know, and because nothing in biology is simple, then you can have viruses that require a combination of more than one of these three factors. So Ebola Zaire virus, first, after attachment to its receptor, it will be endocytosed. And then for triggering of the fusion domain, it requires low pH and proteolytic cleavage, and then an interaction with the co-receptor, which is endosomal. So you require all these three things occurring one after the other. Yeah.
Does that mean they are less virulent because they need more phonation?
You might think they would be less virulent because they need more stuff, right? But these are horribly pathogenic viruses, and they cause systemic infection. They can infect a lot of cells. So I guess that means, okay, endosomal pathway, that's ubiquitous. Every cell can do this. Cathepsin, similarly ubiquitous. The co-receptor, I don't know, but you would have to assume that its expression is not restricted to one particular cell type. So it should be at like wide tissue distribution. And the binding receptor also must be quite widely distributed. So it needs multiple steps, but probably every step is available in a lot of different cell types. Okay, one more thing to think about with relation to envelope or fusion proteins is that something that viruses can provoke or envelope viruses can provoke is membrane is a formation of syncytia. So syncytia are masses of cells that are infected, and their plasma membranes have all fused together. So measles virus can do this, and you can see that in this culture of cells infected by measles virus, you've got non-infected cells here, individual cells, and you've got this big, horrible monster, which is a syncytium. You've got all these nuclei, and all of these cells are fused together. And SARS-CoV-2 was also able to provoke syncytia, and you can see this in some clever system whereby the fused cells become fluorescent. So you can see the alpha and the beta strains of SARS-CoV-2 were more fusogenic. Okay, you got bigger syncytia formed. So that's a particular type of cytopathic effect, which you can see in cell culture. It can be important in cell-cell transmission of the virus. So locally, infected cells can fuse with neighboring cells and infect them, and the infection will spread locally. But it's only going to be possible if two things two conditions are required. So the first one is that the fusion glycoprotein has got to be on the plasma membrane of the cell, which is not always the case. Sometimes the particles are formed in the endoplasmic reticulum and then exported. So the fusion glycoproteins aren't really concentrated on the plasma membrane. So, and the other thing is that it can only occur if triggering is possible at neutral pH. So that's true for SARS-CoV-2 because it's a TMPRSS2 protease which is doing this. It doesn't require internalization. So that's kind of another function of these fusion domains. Fun fact here is there's a human protein cytin-1 which appears to have come from an endogenous retrovirus that has inserted itself into our genome. And anybody heard of this before? What its function is? It's expressed in the placenta, and it has an important role in fusing neighboring cells during the development of the placenta. So we've kind of co-opted this viral fusion domain to perform a function for us. Okay, now it's time to just stop for a bit. I've been talking a bit too long, so think about these things, and I'm pretty sure should be easy. Yeah, okay. Okay, question 1. Membrane fusion triggered by the interaction between a viral attachment glycoprotein and its cellular receptor. Who says yes? Who says no? The answer I'm looking for is no. So maybe I should have had directly triggered. Okay, the process is initiated by the attachment, but the triggering itself is something else. Okay, after attachment. The specificity of the attachment to the host cell is ensured by the high affinity interaction between the attachment protein and receptor. Who says yes? Who says no? Good, you're right, you didn't fall into the trap. Low affinity, high avidity. Fusion domains are involved in silicium formation. Who says yes? Who says no? But the answer is yes, just on the previous slide. Are involved in and required for. Question 4, membrane fusion is accompanied by uncoating. So there's 3 answers here. There's yes, no, and depends. Okay. So who says yes? Who says no? Who says depends? Yeah, you're right. You guessed right. So that's true because it depends on the virus structure, right? So if you've got a virus which is kind of like this, with the genome inside the capsid, and you've got a virus kind of like this one, dengue fever virus, which we saw, and this one will be herpes simplex. You've got the genome like this. Here you have membrane fusion. Decapsidation occurs. Okay, the genome leaves the capsid. Here you have membrane fusion. What enters is the whole capsid and the genome is still inside. So you have membrane fusion first, decapsidation has to happen later. Okay, so the answer to this question is depends. Okay, and it depends on the structure of the virus. Okay, so we have to cover non-envelope viruses in 20 minutes. I think that would be okay. Right. So the example here is going to be the Picorna viridae. So it's big, diverse family, 3 genera important for human infections in any case. Maybe not human, yeah, maybe. So cardiovirus, hepatovirus, so this is like hepatitis A virus, and the enterovirus virus genus is composed of dozens of enteroviruses and rhinoviruses, and poliovirus is an example of an enterovirus species. So all of these picornaviruses are non-enveloped viruses with a T equals 3 icosahedral capsid, and inside they have a plus strand RNA genome, so they're Baltimore class 4, and the overall length of the genome is maybe 7 to 8 kilobases in length. So how do these guys infect cells? So generally speaking, the overview is kind of like this. So first, the viruses will attach to their receptors. For the main group of rhinoviruses, it's the ICAM1 protein. After this, the virus will be internalized, and it's a combination of receptor binding and lower pH, which will cause a reorganization of the capsid proteins, and some of which will form a pore that crosses the endosomal membrane, and then the viral RNA gets released into the cytoplasm. So here, once again, wow, okay, so you've got binding first, then internalization, entry, and then decapsidation as a third step. Okay, so let's have a look a little bit closer at how the capsid is reorganized. So what is possible to do is you can purify infectious virus particles, and then you can treat them with perhaps a soluble form of the receptor, the ICAM1 external extracellular domain. So the receptor will bind, and then you can reduce the pH of the buffer that you have the virus particles in. So you can try and recapitulate what is happening in a cell during infection in, in a test tube in vitro. So people have been doing this for years and years and years. And because cryo-electron microscopy has become more and more powerful, you can get a better picture of the structures of the proteins in the virus particle. So this is data from a paper published 12 years ago now. So this is the outside of a human rhinovirus particle, and these are like cross sections. So as if you've taken this and you've taken a slice through the middle of it, okay? So you can see the protein shell around here, and all this pink stuff, that's the genome, that's the viral RNA in there. So this is at pH 7, and then when you put the pH down to 5, then you see that you get these progressively more and more empty particles. So how is the RNA coming out here? Well, you can see the difference between here and there. You've got some RNA touching the capsid here, and this is at VP2, I think. Here you don't have it, but as the pH goes down, you see some RNA touching the VP1 pentamers here. So if you kind of zoom in on this region, neutral pH, it's like this, kind of closed. VP4 is here. And as the pH goes down, slightly more open. VP4 disappeared, it's come out. And you've got this electron density, which is the RNA that's coming out of here. Okay. So the model is that after you get receptor binding and reduction in pH, the receptor binds at this junction between VP1, VP2, and VP3, and that's going to cause release of this small organic molecule called pocket factor. So this is released, and the whole thing becomes unstable. Okay, so VP4, which has hydrophobic peptides, VP1 has got hydrophobic domains, they're all going to project into the endosome membrane, and that will form a pore across the membrane that's kind of continuous with the particle, but you can't see it in these images here. And that's where the viral RNA comes out. So that's the model for attachment and entry, for example, for a non-envelope virus. So one of the things we talked about last time, and we started with Vincent's little explanation, is how stable virus particles are, okay? So, or the non-envelope viruses, they're generally pretty stable, but they can't be 100% stable, okay? If you've got this fantastically strong protein cage around the virus nucleic acid, they'll never be able to infect a cell, right? Because at some point, the capsid has got to be able to release the virus genome. So what we say is that non-enveloped virus capsids are metastable. Okay, they're stable in some conditions, but they can be unlocked, okay, and they can fall apart. And you have to get the right conditions to allow the virus particle to dissociate. So here, it's the interaction with the receptor and the low pH. And that will work pretty well for respiratory rhinoviruses, because they're infecting cells up here. They do, in the respiratory tract, they don't go through a low pH environment in the stomach. Okay, so for picornaviruses, as we saw, the entry causes the release of the viral RNA and is going to go to the cytoplasm, and that's okay. But for some non-envelope viruses, all the replication cycle is going to occur in the nucleus. So that's true for most DNA viruses, and yeah, so for most DNA viruses, the replication will occur in the nucleus. So that means the decapsidation can't be immediate. It's got to be delayed. So the virus has got to somehow get translocated to the, not transmission, then transport or something. This was a Google Translate and I didn't fix it. So another example here is for adenoviruses. They have got multiple steps in their infectious entry pathway. So the first thing is going to be attachment via the Coxsackie adenovirus receptor, the CAR, and that's going to be the kind of knob domain on the spikes that are on the fibers of each of the vertices of the capsid. That's the first step. Second step, there's got to be an interaction between the co-receptors, which are integrins, with the pentons down here. So it's probably the co-receptor distribution that determines the tissue tropism of these viruses, because you need both things to be present on a susceptible cell. You need the receptor and the co-receptor. So for adenoviruses in the table earlier on, I didn't have tropism for the Coxsackie adenovirus receptor. Probably because the co-receptors are more involved in tropism. So these co-receptors are necessary for the endocytosis. The pH is going to go down. This will partially disrupt the capsid, and you get some viral proteins. I think it's this one here, protein 6, disrupts the endolysosome and allows this kind of like a particle to be released into the cytoplasm. So from there, it's got to get to the nucleus. So it's going to be transported on microtubules. So the hexon protein here is going to bind to, what is it? It's dynein, which is responsible for retrograde transport, right? Anterograde transport is kinesin. Is that right? And the retrograde transport is dynein, this kind of like protein motor that takes cargo and moves things around the cell. So the adenovirus capsid will recruit the dynein motor and take this kind of like train ride down to be released close to the nucleus. And the last step will be attachment to the nuclear entry pore. And it's here where you'll finally get decapsidation and release of the virus DNA into the nucleus. So this is kind of the last slide for today, I think. I've got a few minutes, so I might start the next one. So the nuclear entry pore is, it's also a barrier. It's selective, doesn't let anything in or out of the nucleus. So every virus that replicates in the nucleus has got to have a strategy and a mechanism to get into the nucleus. So influenza virus, it's an exception among RNA viruses. The replication occurs in the nucleus. And the way it gets in is that the nuclear capsids carry a nuclear localization signal in their proteins, so they get imported by the alpha and beta importins. So they just like take the normal import pathway. And the size of the nuclear capsids is small enough to go through the nuclear pore. Okay, because they're these like elongated helical capsids. So small icosahedral capsids can also go straight through the nuclear pore. Okay, so that's true for parvoviruses, which are very, very small icosahedral capsids. They can actually get through here. So then you get the whole capsid is imported, and then finally it dissociates in the nucleus and releases the viral DNA genome. However, for bigger icosahedral capsids, they can't get through. They're too big. Okay, so for adenoviruses, what's going to happen is that the capsid will dock at the nuclear entry pore, and then it will be dissociated by cellular proteins, and the DNA will be imported. Herpes viruses, it's a bit more elegant. So they will dock on the nuclear pore. And if you remember, herpes viruses they have a specific structure of their capsids. So they have a kind of like collar at one of the vertices of the capsid. So this is what's going to dock onto the nuclear entry pore. And then your viral DNA is here. So the docking interaction will open up this pore and the DNA gets injected out into the cell. Okay? So the capsid just stays there, but the DNA goes in. Whereas for adenoviruses, the capsid has got to be like pulled apart. Okay, so that's about the end for attachment and entry. So the main points here, it all starts with a specific interaction between virus attachment protein or glycoprotein and a receptor on the host cell. An important exception is plant viruses. There's no real specific entry receptor for most plant viruses. The structure of the virus particle determines the entry mechanism. Okay, so for all envelope viruses, there's got to be membrane fusion at some point. And for, you have to understand, the three main classes of fusion glycoprotein. And for non-envelope viruses, either at some point there's some kind of portal that's formed that's going to let the virus genome get out, or the capsid's got to be, has to disintegrate. And this will, and of course, this step's got to occur at the right time. Okay, if this happens too early, you've got like premature genome ejaculation here. It's not going to be good for replication of the virus. And then last point, so these receptors, co-receptors, important for virus tropism, except for influenza, because it's the priming protease which really restricts the tropism. So we got 5 minutes. I don't know if it's worth starting. Probably not. I think it's going to be good if I stop. And yeah, I'm picking up some very subtle signals here that tell me that it would be good to stop right now. Okay, so we'll see each other again next week, right? So next week we also have the TDs that start, I think. So if anybody has any questions about the TD work, we can use this like 5 minutes that we got. You can come and ask me stuff, okay? Anybody has any questions about the presentations?